First, we have Dr. Benjamin Freedman. He's worked with stem cells, and he has helped regenerate body parts. Studied that and how mammals do it. It's pretty interesting. You've done a lot of really interesting things. We're really excited to hear about what you're doing. And so we're going to turn the mic over to him shortly. And then we also have Mr. Bill Steele from the University of Washington. He's the director of the Educational Outreach and Communications Program for the Pacific Northwest Seismic Network. And so Bill's going to help us understand how we might plan ahead or think intelligently about what might happen when we have an earthquake here. So anyway, please welcome you both. Why don't you lead us off? Benjamin, tell us what you're working on.
All right, great. Thanks for the introduction. And I've kind of changed the title a little bit to Manufacturing Organs to Treat Disease, more in the business theme. So I'm going to discuss, we really haven't regenerated body parts yet. But we have an idea of how this can work, and we've made some progress. And the key thing to understand is shown in this slide. On the left of the slide, you see a person. And this could be anybody in this room. And what we now know how to do is take cells from that person. And these are shown at the top. These are the adult cells. Now, these are not many body parts. But what we know how to do now is introduce into the cells that are in our body genes that are expressed while we're being developed in the womb. And what this does is it reprograms the cells in our body back to the very earliest stages of development before any of the body parts have formed. And these are called iPS cells, shown on the right. And we can grow buckets of these in the lab. And now the trick is to figure out how to get these cells to change into the various types of body parts. And recently, we've made a breakthrough in our lab in figuring out how to get these to turn into kidney. And that's what I'm going to talk about today. And they don't form whole kidneys, but they form what are called mini kidneys. And these are shown on the bottom here. And we're using these mini kidneys for two things. The first aim is to use them as surrogates for our own human kidneys. Because we can't just take human kidneys out of people and do experiments on them. Well, we can do these types of experiments on these human mini kidneys. And we can figure out which drugs might help heal these kidneys. Now, the second aim is shown on this arrow from the mini kidney back to that original person. And the idea here is that if we can grow body parts from our own cells, we could, in theory, transplant them back into our own bodies, and they would be 100% immunocompatible, meaning they would not require any type of immunosuppressive medications. Furthermore, they could be produced on demand, so there would be no waiting lists for transplant organs. Now, we're not there yet, but that's a long-term aim, and I think making it to the mini kidney is an important step along the way. All right, this slide here is just showing you the mini kidneys compared to actual kidneys. And the way the kidney works, it's a big filter unit, essentially, and it's made of these little tiny filter subunits, which is shown in the center of the slide here, and that's called the nephron. And the way it works is that the blood flows into the nephron, and it's filtered into this tubule, and then there's a very complicated process of taking back all the good stuff and throwing away only the things the body doesn't want or doesn't need. Now, these little tubules are not things that are easy to grow in the lab, and we've had a lot of trouble growing them. But now, using the stem cells, we can make these mini kidneys, shown on the right side of the slide, and these have the same types of cells that are found in the actual kidney. So the red are the filtering cells, and the green are the tubule cells through which the urine passes. And these structures really make very beautiful, intricate, complex subunits that resemble what are seen in the middle of the slide in the actual kidney, same types of cells. Now, it's not a perfect copy. There's still a lot of things missing, but it's amazing to think that the structure shown on the right, that mini kidney, was actually once a skin cell. It was taken from a person, it was reprogrammed back to a stem cell, and now it's totally changed its fate into kidney, into growing this complex unit. Now, in the meantime, we can't transplant these yet. It wouldn't be safe or responsible. But we can use them to study disease. So we wanted to know, could the mini kidneys actually get disease? And the disease I'm going to focus on is called polycystic kidney disease. It's extremely common. 1 in every 500 people on the planet has this disease. Chances are somebody in the room has the disease. The way the hallmark of the disease is shown on the left side. The kidneys swell up, and they have these large balloon-like sacs full of fluid. And eventually these balloons, which grow from the tubules, crowd out all the healthy tissue. Now on the right side of the slide, I'm showing you a picture I took from a Coldplay concert several months ago. It's CenturyLink Field, sold out show. So this is about, I think about half the stadium shown in the image. So that captures about 35,000 people. Now if you do the math, that's about 1/400th of the world's population with PKD. We don't have an effective drug for this disease, but if we did, we would be curing 200 stadiums, football stadiums worth of human beings. Now just to show you whether the organoids, the mini kidneys, can actually get PKD, we've figured out a recipe for this, and I'll just get to the point here. If you look at the bottom of the slide, you can see that the kidney organoids, the mini kidneys, without PKD on the left, and the ones with PKD on the right. And you can appreciate that the ones with PKD, and we've only changed a single gene in these mini kidneys, are swelling up like balloons, just like what the disease looks like in the actual kidneys. And these swollen structures, they grow so big that we can actually see them by eye. We don't have to look through a microscope. So they're about 1 centimeter in diameter, and sometimes larger. So this is a picture I took with my iPhone, and you can see we're zooming in here on these diseased mini kidneys, and they're swelling up with this disease. So that's what we call disease in a dish. Now, what we'd like to do is now test huge libraries of drugs to test which ones will actually cure this disease. And these are chemical compounds that can be synthesized, but we don't know which one might actually be a good drug, a lead candidate. So what we're doing is we're having, instead of having students make the mini kidneys, we're having robots manufacture them in the lab en masse. And these are pictures of this here. On the left, this is a little robot that's printing out in these tiny little micro wells mini kidneys. And on the right, we're zooming in, and you can see how in each of these little wells, it's peppered with these little mini kidneys. And this is how we're now being able to look at different drugs. So the robots are helping us do this at many different doses for each compound, and many different compounds at a single time. And this is just one example of where we profiled 8 of these possible drug candidates at different doses, and we've seen that this one called blebbistatin, shown in red, at a certain dose, greatly increases the size of these mini kidney PKD cysts. Okay, so we found a pathway that seems to be involved in the disease. Now it's doing the opposite of what we would really hope to see, which is to rescue the disease, but it's a start. So these discoveries perhaps would be better explained by someone who's a non-scientist. So luckily we had the news come into our lab recently, and I just want to show you this little clip. And even if you haven't listened to or understood any of what I've said so far, the news will maybe help
New attempt robots and research underway right now at the University of Washington may someday save your life. Scientists are using those robots to help make thousands of miniature kidneys every day with a goal of curing kidney disease. Congressman's Joanna Small went inside that lab to find out how it works.
This is our main lab.
Benjamin Freedman is a scientist, but he might not be if it weren't for his uncle.
The thing that really got me into it is actually that my uncle had kidney disease at the time. And I was thinking we've got to use these stem cell technologies to find a way to find better therapies for people with this disease.
So for the last decade, Freedman has been researching kidney disease.
They have to be cultured.
And in 2015, he had a breakthrough at the University of Washington using microscopic stem cells and with the help from robots. He grows kidneys, thousands and thousands and thousands of kidneys.
There are 384 stem cell samples in each one of these trays. On the other side, you have a tiny pin with an even tinier opening. What you see the robot doing is taking a compound and adding it to each one of the stem cell samples, and that is how a mini organ comes to fruition.
They're faster, they can handle larger numbers, and they're more accurate than human beings. Once that transfer is done, then we take those and they go into an incubator.
It takes 3 weeks to develop a mini organ, and they're so small you can only see them under a microscope that magnifies the images by roughly 1,000.
We zoom in. Each one of these wells contains several of these mini kidney organoids, these small structures.
So what do Freedman and his team do with them? Well, the answer is twofold.
So we're using these little mini organs for what we call clinical trials in a dish to try to figure out what drugs can help cure different kidney diseases.
For example, these kidneys are visible to the naked eye because they've been injected with a disease that makes the organs swell.
These have not been treated with drugs. We're just letting them grow to study how they behave.
The other application may be even more impressive. The wait for a kidney transplant is long and arduous, and even those lucky enough to receive a compatible one.
Eventually they do wear out. It's not like if you get a kidney transplant, it lasts forever. You have to get another one after about 10 or 15 years because your body attacks it no matter what you do.
But this technology is essentially growing a kidney just like the one you have, a clone, only healthy.
Our bodies will be very happy with our own cells. They just will reject cells from other people.
They have done some implanting in animals. Have you had positive results?
Not to this point. So
Yet, Freedman says that's not so far off.
if we do well, we could potentially start trials in humans in about 10 years.
That's about the time his uncle will need a new kidney. He waited 5 years for his first one, and that was 5 years ago.
Oh, if this works, it will change for him and for many others. There's so many people who are in need of these transplants.
At the University of Washington School of Medicine, Joanna Small, Cairo 7 News.
Okay, so that's all I really wanted to say. I'll just leave by leaving you with one thought, which is that this is not just kidneys, but you can actually do the same type of technique for any organ or part of the body. These cells are that powerful. And right now it's a matter of just figuring out how to do that. And I'll just thank everybody in our lab who was involved in this project shown on this slide, particularly Nellie and Stefan, who are scientists in the lab. And we've had great funding support from foundations as well as the government and from the University of Washington itself, various divisions shown here, as well as the Northwest Kidney Centers. And thank you for your attention.